My name is Maristella and I'm a PhD student in pharmacological sciences at the University of Padua, Italy. Currently, I'm also a visiting PhD student at the University of Washington, Seattle, USA. I started getting interested in science when I was young because my mom is a math teacher, and when she gave a private lesson to students, I was sitting next to her copying numbers or equations. And then when I grew up, I became more passionate about science and I decided to study pharmacy at the university. I decided to focus on diabetes because it's a global burden and it's one of the leading causes of death worldwide. Several research groups are studying the molecular mechanism involved in the pathophysiology of diabetes, but I feel my research group is different from the others because we have a cardiovascular perspective. We are trying to discover novel signaling pathways and early markers of diabetes, starting from calcific aortic valve disease. Aortic valve calcification is the most common heart valve disease in the Western world, with a prevalence of 12% in the population over 75 years old. To date, no pharmacological therapies have been approved for the prevention or for the treatment of aortic valve calcification. Diabetes can contribute to the development and to the progression of aortic valve calcification, but the molecular mechanism involved in these pathophysiological processes are still not completely understood. So that's why the aim of my research is to understand which are the critical factors leading to aortic valve calcification in diabetic patients. Our lab has a very unique approach because we are using as in vitro model cells from non-human primate. Compared to other animal cells, these non-human primate cells are more similar to the human one, and for this reason we can use probes tested and approved for human genes for our in vitro model. At the same time, we are also using diabetes models to have a better understanding and to discover novel molecular pathways involved in the pathophysiology of calcific aortic valve disease. Applied biosystem technologies have innovative tools that help me analyze the gene expression profile of our in vitro system. And in particular, we are using their machine, their probes, and their reagents to perform real-time PCR in order to assess the expression levels of several genes in aortic valve cells after different treatments. We are using valve interstitial cells, which are the predominant population of cells in the aortic valve, and these cells can change their phenotype in response to several external stimuli, playing a fundamental role in both the development and progression of calcific aortic valve disease. The analysis of their gene expression profile with Tachman gene expression assay can help me understand how the disease develops and which are the key or critical factors for its progression. And it's particularly useful for us that we can use the Tachman gene expression for human genes for genetic material from non-human primate cells. As a young researcher, I hope that in the next future some promising treatments can give positive clinical outcomes and can be approved for calcific aortic valve disease, especially in those populations at risk, including diabetes subjects. Most of the research in my field is very recent. We don't have a deep understanding of the molecular mechanism leading to diabetic aortic valve calcification, and so I hope that in the next years we will be able to find some therapeutic targets for novel pharmacological treatments.